Thanks so much for taking the time out of your schedule to come out over. So I'm very, very pleased that we have Dr. Carolyn Wagner here today. So she's an assistant professor in the Department of Biomedical Engineering, also a member, or bioengineering, sorry, also a member of our Center for Climate Change and Health. And it's really, I think, for students that are here who are working in population health, I actually think you're going to be really interested in this talk, in part because she has a lot of training in population disease modeling. So has worked on that. She has degrees coming from MIT and Princeton, and is going to be talking about some of the work that had 3 publications in Science. So, I think this is really, really cutting edge stuff, and I'm delighted that she's here today to present for all of us. Caroline, over to you. Or Caroline, sorry, Caroline. I have to do the share.
Thank you very much for introducing me, Phil, and it's really nice to be here. I'm sort of slowly getting more involved with this side of the campus and managing to get more involved with the center and obviously the EPOH and School of Population Global Health. So I'm really happy to give this talk. Oh yeah, that's perfect. Okay, so I'm going to talk today about my little thing in the middle. Hopefully it'll go away. My research program in engineering that basically combines my training working with complex fluids, and you'll see why that's important in a second, with disease modeling at the population level. But I'm going to focus on what I'm thinking about in the context of climate change, because obviously that's the big point for the center. So I don't have to sell to anyone that humans have played a role in how disease is spread. This is from a really nice review article that I got to help write that's become quite well cited in the context of how infectious disease will change sort of as global factors change, including climate change. And what you can see is, there's been a whole bunch of mechanisms through which diseases have been moving around as a result of human activity. It used to be more localized, sort of due to restrictions on intensity of movement, but obviously with plane travel and huge increases in global travel patterns, diseases have been able to spread much more rapidly. It's encouraging to see that overall we've seen achieved declines in terms of the burden of infectious disease, let's say over the last 20 years, as we've had improvements in sanitation and access to healthcare and things like that. Although we can't ignore that there's still a substantial burden that remains, especially as I'll point to in sort of developing or low and middle income countries where, if we just think about, Canada or the US, what are the major causes of death? This is a little outdated right now. This is 2016. We see a lot of blue on the left plot here that's coming from mostly non-communicable diseases. And we could think, oh, well, we should worry a lot about stroke and stuff like that as engineers thinking about interventions. But obviously, when you shift the table and think about what has a lot of impact in other parts of the world, communicable infections, including respiratory infections, still present a very important burden for disease. So this is again from this review paper. It's sort of pointing to different ways that humans can be involved in this process. And I'll say humans again in the context of climate change, obviously owing to the human contribution to that factor. And so this review covers a broader set of concepts, including sort of how demographic changes are important, and it breaks it down to thinking about sort of how it can impact different aspects of infectious disease transmission, notably where pathogens will emerge, the actual dynamics of person-to-person spread, and then how easily things can spread at the global level. So with demographic change, with sort of increasing human expansion into historically rural or animal wildlife territory, we can increase contact with reservoirs for infection. Increased population density can impact how easily infections can spread, and sort of, as we were saying, increased travel can increase the range and speed with which pathogens get around the world. Technological improvements affect things in across the board too, obviously, in good ways too, like having vaccines obviously can be very beneficial in terms of this picture. But climate change to me is interesting because it can impact stuff on multiple levels. So first of all, as we'll talk about, we can shift the range for reservoir species for a disease can exist. Then we can also impact the way that it will actually transmit and susceptibility of hosts. And that's what I'm going to spend most of the time today talking about. And then particularly in the context of vector-borne disease transmission, can have a huge impact on the range of the vectors that will transmit the infection, which means the infection itself. So this is the image that's presented in this review, but I think there's actually more ways that climate change has an impact when we think about infectious disease transmission. And this comes from my training in biofluids and the work that I do sort of in host. There's evidence suggesting that with climate change, we can have pretty substantial impacts to the host susceptibility to infection in the first place. One project that we have, I'll talk about a little bit, is how air pollution can impact potentially the severity of respiratory infections. It can certainly increase pre-existing respiratory infections like COPD and asthma, but whether or not it could enhance, say, an infectious disease is something that I think needs to be looked into more carefully. And then there's also a lot of evidence that climate impacts your baseline immune response, which means that with climate change, we could see this be modified. So it's interesting because in the context of vector-borne disease transmission, we actually understand more in some ways about how climate plays a role. Here's a picture sort of showing which aspects of this are impacted by climate. So right away off the bat, this concept known as the extrinsic incubation period, which sets the timescale between when a vector takes a blood meal from an infected host and then can go on to transmit the pathogen to a susceptible host. This is climate sensitive, and there's been a lot of really amazingly detailed studies untangling why this happens. It's a lot easier to work with tons and tons of mosquitoes in a lab environment than it is with people. So you can put all these mosquitoes in different temperatures, and you can see that pathogens will develop at different rates in the mosquitoes. The mosquitoes themselves will develop at different rates. And then, of course, it's not too much of a stretch to believe that precipitation will play a role in this too, because they like to lay their eggs in water reservoirs. And so there's long been this connection between, let's say, mosquito-borne diseases and rainfall patterns as a result of this. So in some ways, the mechanisms are a little bit more easy to disentangle. So this is, for instance, the way that people who work in infectious disease transmission can incorporate climate in quite not straightforward, but let's say a clear way. They can say, all right, if, let's say, mosquito life cycle depends on temperature in this way and pathogen reproduction depends on this way, then what people in this space have done, and this is a preprint from Erin Mordecai's group at Stanford, she does a lot of work in this vector transmission space, is they can say, okay, assume I input these values for temperature and precipitation. I can run my models and see what I'll expect for burden of disease. But then I can use the change projections for temperature and precipitation that the climate scientists give us, run the models again and see how burden changes. So that's what's being done in this paper. Of course, when you use a projection for climate, you have to assume some pathway for what climate's going to look like. So you all probably know this. Typically, you make assumptions about whether you're in a pathway that assumes we do mitigate greenhouse gas emissions substantially or not. So the top one is a low emission scenario, so that's a more optimistic pathway, whereas the high emission scenario is less optimistic. And what this group saw, this was on dengue transmission across the board. It doesn't look great, so you see a lot of red and purple, which means increases in dengue burden. What I find a little bit terrifying is that in some places where you see the decrease in dengue burden, that's actually because the temperature is now so hot that the mosquitoes won't be happy reproducing anymore. So that doesn't really seem to be good news, because presumably humans won't be happy under those conditions either. But in some ways now, when we think about respiratory infectious diseases, the picture becomes a little bit less clear, even though we see strong evidence of seasonal cycles. So this plot is from a review article looking at an influenza transmission in many places around the world. And I find the plot a little bit tough to get intuition from, but essentially what's being shown here is the timing of the peak month for the primary influenza peak in different parts of the world, so plotted by latitude, against what month that corresponded to for the corresponding temperature or precipitation or specific humidity level. And the trend that you see largely, and this is pretty well known, is that at high latitude regions, influenza tends to coincide with low temperature, low specific humidity months, wintertime months. But sort of one puzzle that's always made people sort of wonder more about this mechanism picture is that in tropical latitudes or low latitude regions, there actually seems to be more influenza during rainy season or high precipitation months. So that's sort of an interesting puzzle. We see evidence of seasonality though. They can also probe seasonality more directly with animal studies. So this is a very highly cited study where they basically put guinea pigs. You can't use mice as a influenza model because they aren't susceptible to influenza in the human strains that we want to look at. So guinea pigs tend to be the model system that's used, and they just put them in one cage and then the other guinea pig in the other cage so that they know that they're not like directly coughing on each other, and they can just look at transmission across larger regions, which you would think would be due to aerosols and things like that. And they can look at how much the originally healthy guinea pigs get sick, and largely what they see is that cold and dry conditions favor transmission. This is RSV, so obviously, especially in the fallout of COVID, we saw a huge impact on RSV dynamics around the world because of the NPIs in place for COVID. So it's very problematic if you're very little, an important cause of death globally. And here, this is from my colleague Rachel Baker at Brown, who's a master of figure creation. So what you're seeing here is by geographic region across Mexico and the US, the timing of the onset of an RSV peak annually. And what you can see is it shows it's very strong correlation with geography. So the onset week shifts as you move from the southeast up to the northwest. And actually the patterns of cycles, whether they happen annually or biennially, also seems to have a geographic correlation. So seems like there's a lot of evidence that respiratory infections are seasonal and impacted by climate, which means that if the climate changes, these patterns that we know can potentially change as well. But disentangling what those mechanisms are is less straightforward. And that's something that we try to work on with what we do experimentally. So typically when you look at papers, effectively what's done is they take, scientists will take a time series of cases, obviously beneficial to work with diseases for which you have a long history of case data. It's challenging during COVID because for an emerging infection, you don't have a nice long time series to do these manipulations on. So it's hard to come up with these relationships. And then essentially you're regressing some measure of the transmission against the climate variables that you typically do have very good access to. Typically you'll do this with something like an SIR model, which you probably in this audience come across before, but essentially it's a ODE model, a compartmental model that thinks about the world as either susceptible or infected or immune to an infection, and thinks about how people move between these compartments due to transmission. There's some math to make this at a discrete time scale to match the data that you typically have. But then your goal is to essentially come up with some regression expression between your climate variables and something related to the transmission term. Then you can go ahead and plug in new temperature or precipitation or humidity data and back out some new prediction for transmission. But there's a lot of issues with the approaches based on this. So first of all, we typically have pretty strong spatial biases for where we can get good epidemiological data from. Like I said, if you have an emerging pathogen, this approach doesn't work because you can't really do any of these regression-based approaches. We're also pretty quickly entering into climate regimes for which we don't have a lot of historical data, which means we'd be moving towards something like extrapolation, which is not really very sound. And then also climate variables tend to be very correlated, so which is not a helpful thing when you're using approaches like this. So why does it matter from my perspective that we can understand more about these mechanisms? It's that with climate change, we expect that the timing, but also potentially the intensity of outbreak patterns will change. And this is important because one of the big jobs for public health agencies locally is capacity planning and resilience. And so if you think, okay, I need this many ICU beds because in influenza season, I typically see this many patients, and then all of a sudden that number doubles or something like that, that will be very problematic. And maybe it would be somewhat problematic in a place that has a lot of healthcare resource access, but if you're somewhere with a more fragile healthcare system, it could be very, very problematic. So from my perspective and from a lot of people's perspectives, coming up with sort of, let's say, first principles understanding of why this is happening, as opposed to relying on sort of more observational correlations, is really important. And I also think that if we do this with respiratory infections, we could extend this to infections that transmit in other ways, but still show climate signatures. So for instance, cholera, we think of as a water-borne infection, but it's long been associated with El Nino phenomena and rainfall patterns. So again, if we can turn what I'm going to show you today, which deals with aerosols, into just a water bath, right? Change the temperature and put the bacteria in there, maybe I can apply this to other contexts as well. So to do this, I think a lot about mucus, probably more than the average person. I'm going to convince you today why it plays this climate-dependent role, both in what it does in the body and what it does outside the body. And what I want to convince you of is that if we can understand more about the dynamics that happen in the body at the in-host level, we can actually gain a lot of insight into what's going to happen. So one story, maybe just to pique your influence and help you understand why I care so much about mucus, is the influenza virus, which is very interesting in terms of its structure. So it has two surface proteins that are really important in terms of transmitting between hosts. It has hemagglutinin, which binds to the receptor that needs to get into the cell, but then it would be stuck if it didn't have its partner neuraminidase that could come along and chop off the connection so it can actually move in. But what's interesting is it's looking primarily for sugars called sialic acids on the end of cells, but mucins, which are the big component of mucus, have a lot of the same receptor on them. So it's been hypothesized that maybe your mucus is kind of acting as a decoy to trick the influenza into thinking it's found a cell, but actually it's just mucus, and then it would immobilize it and sort of take it out of the game in terms of infection. And what I find really fascinating is that the type of sialic acids that you have varies along your respiratory tract, also between species, and different influenza viruses show tropism preferences for different types of sialic acids. And then this paper did a really nice example of how closely we can relate the in-host picture to what happens at the population level. They built a model for the respiratory system, which is what's shown up here, where they basically looked at different clearance rates of mucus and pathogen reproduction rates in the upper respiratory tract, which typically, if you get an influenza infection, it'll transmit more easily, but it will be less deadly compared to the lower respiratory tract, which is typically associated with very fatal infections. And they were looking at how all of these variables that happen in host, I'm trying to use my mouse here, but I don't know what's going on. In the black boxes, they were trying to look at how everything that's happening in host, from which they could extract things like the amount of virus over time, because they're literally modeling like cell to cell viruses reproducing in these cells can be connected to the population level. So typically you would say, okay, let's say person X has twice as many viruses in them as person Y, maybe that means they transmit twice as much as person Y or something like that. Or if they have way more virus, they're going to die more likely than person Y or something like that. So this is how you connect the in-host to the population level picture. And they found some pretty, from my perspective, fascinating results that depending on pre-existing immunity to the strain of the virus that they were considering, which would sort of distinguish a new epidemic strain of influenza compared to one that sort of more circulates more endemically or say on an annual basis, the tropism for where the virus preferentially was infecting changed. And there was more tropism for the upper respiratory tract when there was pre-existing immunity, which is sort of consistent with this idea that the epidemic ones, which will cause a lot of fatality, might be targeting the deep lung regions. So hopefully now you're convinced that something about the respiratory tract matters, and thinking about what happens in the body is connected to what happens at the level of populations. So I'll tell you a little bit more about this detail of what happens in the body. So this is my mucus 101, what is mucus picture. Mucus person here, and mucus coats every non-keratinized wet epithelial surface of your body. So everything from your eyes, your tear film, your saliva in your mouth, respiratory mucus in the airways, your stomach is lined with mucus so you don't auto-ingest yourself, the female reproductive tract, all mucus of different types of proteins. So I know this is not a biochemistry audience, so I'm not going to belabor this aspect too much, but it's proteins basically that make up mucus. That's what I show here is the little squiggly guys, and they have a lot of sugars on them, and it makes this thick, gooey substance that we are all quite familiar with. And their job in the body, let's say if you think about the airways, is to act as a filter. So I draw the analogy of a soccer net. You could think about these proteins in mucus essentially forming the net. And if a soccer ball tries to get through, let's say a pathogen or something that's too big compared to the net formed by these proteins, it gets stuck. So it can exclude things on the basis of size, but it also can stick to things. So let's say you're a ping pong ball, you would think you can get through the soccer net, but if you're painted with the wrong thing, you might get stuck, for instance. So it's really versatile and really sticky. And for engineers who work on like drug delivery to the airways, they're like, mucus is frustrating because they're trying to get something through and it's so good at preventing things from getting there. And this in-host ability, so this isn't even like the sneezing, we're not even at the sneezing picture yet. This seems to be climate sensitive. So there's evidence that when, for instance, you're in a cold or dry environment, the amount of mucus that you secrete is changing, and also that how readily you're clearing it is changing. So part of the picture is, okay, mucus is trapping pathogens, but then you're constantly, as a result of small cilia on your cells, like it's called the mucosalivary escalator, that's clearing mucus out of your lungs and you're swallowing it. So you're getting those pathogens out that aren't supposed to be in your lungs in the first place. So there's evidence that that whole picture is potentially sensitive to climate. And mucus is amazingly good at telling what is in there. So for instance, this is some old experiments that were done in my old PhD group, where these are tiny little peptides, but you're seeing the colors on the right, like 10 nanometers, smaller than you would expect any pathogen to be. And the really interesting story is they took two peptides that were the same charge, but they just played around with where the charges on those molecules were located. So one of them was like a block, positive, negative, and the other one was positive, negative, positive, negative. So just playing around with the way they're decorated. And so the two on the right really matter here. The one that has the block, it's actually enhanced in terms of how much you can get into the mucus, whereas the one that has the alternating, it's sort of agnostic to the presence of mucus. So somehow just even distinguishing the arrangement of what's on the surface of small molecules will impact the type of way that mucus presents as a barrier that it does. And this is relevant to pathogens, for instance, coming back to influenza, because there's been a lot of work looking at how these H and N proteins that I was talking about are arranged on the surface. And some people think it's quite polarized. So it's sort of a block of H and a block of N that would actually let influenza sort of walk through mucus, as opposed to just relying on the random slow process that drives most of biology, which is diffusion. So some people, yes, I think I'll skip over that, but there's also, it's not just influenza, it would be sort of a flu-only talk if that were the case, but so this is a human coronavirus, OC43. It's sort of one of the ones that we think has been circulating for a long time, one of the sort of seasonal cold culprits. It also can be inhibited by the sugars on mucin, so it's not just influenza. There's been some nice work showing that. And from pharmaceutical standpoint, it's interesting that there seems to be some evolutionary conservation between the ways that flu viruses and coronaviruses target different sugars on mucins that maybe makes it hopeful that one day we can come up with sort of a pan respiratory virus vaccine or something like that because of conserved activity on these viruses. I'm not suggesting we're close to that. I'm just saying there's evidence that comes from the sugar picture on mucins that suggests that this might be possible at some point. So part of what we do is just a lot of work looking at what do the different viruses have on their surface? How do they interact with mucins in that way to come up with a better picture for how they could move through this barrier and cause infections? And so some of the work that we do is exactly that. So my PhD student Ishita builds essentially like mock viruses. So what we're trying to do, and I have a pretty new lab, so a lot of what I want to do is work in progress, but we're developing ways to produce little molecules that were essentially all we could do is change what's on the outside of them to represent different viruses. So for SARS-CoV-2, you put spike protein on there. For influenza, we put H and N, things like that, and then compare how they move in mucus. What do we use for mucus? Sometimes we use mucus, but it's typically hard to source and it varies a lot between people. The other thing that we use is we purify those mucins out. So that's actually the sort of more reliable material that we use, either from animal sources, from previously sacrificed animals, or we spend a lot of time salivating into a tube to collect a lot of saliva, which we can then purify. It's quite a long process because you really only want to get those super big proteins out at the end of the day. So there's a lot of purification involved. And then we can use that building block and sort of reconstitute mucus to how we want. We can mix it at different ratios, let's say, or cystic fibrosis. There's a lot of DNA in there that shouldn't be in there. So we can throw some DNA in to come up with disease models. And so the goal is then to look at if there's differences in how different viruses actually get through the mucus in the first place. And a lot of modeling to go along with that. So Leo, my postdoc's just been building models essentially for how trajectories of these different pathogens would change depending on if they stick in different ways to the basic components of mucus. We're also trying to work with cells, which is a new direction for me. I had no cell training in my PhD or postdoc, so it's been a steep learning curve. But there, our goal is to actually recreate, this is called a microfluidic chip. So basically, it becomes really hard to work with fluids at really small length scales. So you resort to doing everything in super tiny little channels, which are not quite micron size, but like the biggest feature would be a millimeter, let's say. And we can create little channels that are lined with cells, and we can flow stuff in there. Eventually it would be just aerosols that we're interested in, for instance. So in context of the air pollution project, our goal is to grow cells, for instance, and then expose them to different types of air pollution and see how that impacts their ability to produce mucus. Because it turns out if you just take cells from the respiratory tract and put them in the right environment so they're happy, they'll just like grow mucus and they'll beat their cilia and they'll do all the things that they could have done in the body. So we can essentially achieve a little airway system in our microfluidic chips. And then our goal is to actually look at the sort of kinetics of how does something that we're aerosolizing get to its target site. We want to sort of include the branching structure of the lungs and all that to make that more relevant. And yeah, so we're looking at that. And this is just another aspect of that project that my student, my great master student, Arian, has been working on. Okay, so that's the in-host picture. Maybe the more obvious connection to climate is actually what happens out of the host. And we know that there's different ways that infectious diseases can spread. This was a review article sort of from the peak of COVID that sort of explain to people the difference between a foamity infection where you sneezed on a door handle and then I touched it and then touched my eye or something and inoculated myself, or a big droplet when I'm talking versus aerosols, small droplets. There's a lot of controversy in this space about whether we really need to be talking about aerosols and droplets in the first place, and we probably don't. There's not really a true size distinction that really stratifies them in the way that people present there as being. So let's think about anything that's coming out of your mouth that's small. I'll use the word probably particle and droplet interchangeably, but I just mean anything that you're producing that's going to go into the air. The lung is where all these droplets are coming from. So here's sort of like a high level picture of what you've got going on. You got the alveoli, which is where gas exchange happens, tiny little sites down below. And then you've got conducting tree of the airway, and then your face region, your nose and your mouth. And for someone who's interested in fluids, this is like a wealth of information to think about the droplets that we're producing, because this system is so interesting from a fluids perspective. So this tree is the Weibull model for the lung, and it basically says you have your trachea, which is like your top generation of your tree, and then it branches and branches into 23 generations, I think. So in the upper portion, you have a lot of the cells that make mucus and contribute to this escalator motion that clears mucus. So, okay, that's sort of interesting, but then as you move deeper into the lungs, those cells are not there anymore. So there's probably not a lot of mucus and there's not a lot of clearance, which is maybe interesting from sort of the deep lung infection perspective. In your mouth, you've got primarily saliva, which we know you collect a lot of it, and your different glands secrete the mucins that make saliva at different amounts. I spend a lot of time measuring the mechanical properties of fluids. So this is actually saliva being stretched between a device, and I'm trying to get some measure of how stretchy it is. Spinnable is the more technical term. And we use measures like that, as I'll show you later, because that plays a really big role in how that fluid will break up when we spray it. These properties of the fluid have a huge impact into the size of droplets that will be produced when you cough, or other mechanisms for fragmentation. Just for fun, I left, actually not for fun, I think I forgot and left the saliva on my desk for a day, and it actually has no ability to make this thread after a period of time. So we actually had some fun writing a paper explaining how that the protein structure would change over time, which could contribute to some of this. Okay, so that's what's happening like in your upper mouth nasopharyngeal region. You got saliva, then you've got mucus as you move into say the first 16 generations of the lung that's being cleared continuously. Then you have the deep lung where we think it's actually mostly a fluid called surfactant. And a surfactant is a molecule that likes to have two parts to it. One that likes water, so it puts that part on the water, and then the other part doesn't like water, and so it sticks up away from the water. And the job of surfactant is actually to decrease what's called the surface tension of your lung fluid, which allows your alveoli to stay in an open configuration. And otherwise, if you didn't have surfactant, they would just sort of collapse because the pressure required to keep them in that configuration would be too high. So in terms of interventions, actually, it's sort of interesting from a pharmaceutical drug perspective. Again, like neonates who are born, if they go into respiratory distress, it's often because their lungs aren't producing their own surfactant yet. So you have to administer some surfactant to allow them to keep their alveoli open. Okay, so what this means is, as a complex fluid person, hopefully I could do a better job characterizing the mechanical properties of all these fluids. Are they squishy? Are they stretchy? Are they slippery? And then what that helps us think about is what kind of droplets are being produced when people talk or do other kinds of activities. And so there's amazing work looking at the types of droplets coming from all the different parts of the lung. When you speak, it's sort of gross, but you actually form these like saliva filaments over your lips that then will just break up into like big bags and droplets, versus your vocal folds are actually vibrating when you're talking, and there's almost like a little ping pong effect where you're sort of bouncing little droplets off. Those are saliva based primarily, but then when you cough, that's deeper lungs, that's mucus that you're shearing, and there's a lot of interesting fluid mechanics to come up with. It's called a fluid instability, where you have a flat layer and then you do high-speed flow over it. Sounds like the ocean on a windy day. You get waves coming from the fact that you've got this high-speed airflow over, so it causes a different mechanism of breakup. And then deep in the lungs, actually, you temporarily can have closure of some of your airways, and then that process of them reopening will also create droplets. And so what people have been able to measure, and I find these data amazing, is the density of droplets that are produced by size for different activities. And what you can see is breathing, which is the green plot here. You get a lot of small droplets, because that's primarily coming from this deep lung mechanism, which means it's probably a surfactant heavy fluid. Which could impact, if the virus is, let's say, in your deep lung or not. Would you think that you're actually expelling it or not? And then as soon as you start talking or shouting or vocalizing, you get bigger droplets show up that aren't there before. That's because of this sort of film effect. So those are primarily saliva-based droplets, and you produce a lot more when you sing, for instance, or scream than if you're just talking. And I have a colleague who measures this directly. Like, she'll get people to come in and say like P sound versus a A sound, for instance, will produce different types of lip motions and droplet production. So from that perspective, it's interesting, but also interesting when you think about where the droplets are originating from and how many are being produced. So this is just a demonstration of how much the fragmentation process will change as the fluid composition changes. And so here, my former student Nicole took some pictures of spraying different solutions. This one down on the bottom right is actually our purified mucin, and it forms pretty big droplets, versus if she sprays just buffer or water, which is the leftmost picture, you essentially just get a mist of droplets. So the size of and distribution of the droplets will change pretty substantially. And that will impact how things will evaporate, right? Does that mean you're going to produce all tiny mist of droplets that will stay suspended forever, or these big droplets that will just fall to the floor and impact person-to-person spread? But it's harder to come up with these models for how things will evaporate when you think about these big proteins that are in the droplets. There's been a ton of papers saying if everything was just pure water that was coming out of your mouth, what would be the size of droplets, how quickly would they evaporate? But we think that the picture will be different with proteins that will impact this essentially fluids and mass transfer problem, but that gets pretty complicated pretty fast. This then impacts where something will deposit in a host. So this is it's based on modeling work at the end of the day. But essentially it looks at, there's been a lot of mostly numerical models that are created for the lung, and it'll look at if we flow in particles of different sizes, where do we expect them to end up as they're moving through this tree? And so here what you're seeing is of the total fraction of particles of different sizes, do they end up primarily, let's say, in the alveolar region? There you can see really the only peak is for very, very small particles. So if you're a big particle, your chance of making it to the deep lung is very, very small. Versus if you're a big particle, it's very likely you'll impact somewhere up high before you even have a chance to get any lower. Okay, so I think that whole picture hopefully is convincing enough that mucus will play a role in terms of how we fragment droplets, and we should care about that because the size of droplets that will be produced will impact the different modes of spread. Is it going to be tiny aerosols? Is it going to be large particles that might sort of pose less of a long-term threat to transmission? But there's also the question of the virus itself in the particles, and that's something that we're trying to study as well, because there seems to be evidence that the survival of these pathogens in droplets is directly impacted by climate as well. So there's been groups that have tried to do this. Honestly, most of the data that people report these days comes from the Harper and Tyrrell papers from 1962. So there's probably room to improve with updated experimental tools, our understanding of this phenomena. But essentially what people will do is take a virus of interest and try to make some droplet out of it, and then expose it to different climate conditions and see how viable the virus remains over time. So there's evidence that there's climate sensitivity, that this is impacted by temperature and humidity, which I'll explain more in a second. There's also evidence that the virus being in a droplet that contains mucin or doesn't contain mucin will change this picture. So if you just put the virus in a buffer that has some salts in it, the way it survives as a function of climate is different than if you put big proteins in there, which makes sense because they will impact the whole evaporation process and the way that the virus will be interacting biochemically with its environment. This paper I love, they just took some money and cut it up and put some mucus on it that they had inoculated with flu. And they showed that they could collect viable flu for longer if they put it in mucus before they, I don't know why they cut up the bank. Oh no, I guess it's because they wanted to put it in a test tube, but I thought it was great that they made such a sort of, the picture was really like, if you get a banknote from the bank, like how likely, I guess, are you to pick up an infection or something like that. And so why does this happen? So there's a lot of thought that that temperature will cause what's called inactivation or protein denaturation for the virus. So at the end of the day, viruses are just a bunch of proteins that are ordered together in a very, very controlled way with some genetic material inside. And if the structure of the protein changes, so this is some nice numerical recreations of actually just the spike protein for SARS-CoV-2. The yellow X is the ACE2 receptor on the cell, and the color regions are the different domains of the spike protein. And what you can see is if the two little blue hands are like this at the end of the protein, it won't bind to ACE2. It's only if they open slightly, then it will be able to bind to ACE2. And this is just a protein folding problem at the end of the day. They need to be in the right configuration. So the thought is if temperature is denaturing the protein in some way and causing that structure to change, then it won't be able to, interact with its target or something. You're not killing it in the sense because viruses aren't alive to begin with. You're sort of just denaturing or changing its structure in some ways. Humidity also seems to be a factor. So this is sort of looking at how potentially humidity could impact virus survival. There's been, I would say there's more sort of theory and hypotheses in this space than concrete evidence for now. So it's something that we're working towards. But basically the idea is if the droplet completely evaporates and the virus is just in sort of its naked state floating around like a little blob of solids, it will be which would happen in low humidity environments, that will impact its survival compared to if it's staying in a water heavy environment, which would be the case at more humid environments. So we're trying to make contributions to this space in my group. This is what my student Mark is working on right now, and we're just at the early stages of this project, and hopefully it will pick up over the next couple of years. So for now, we've only been working with bacteriophage, which are viruses that infect bacteria. So it's sort of a safer starting system as we work out all the details before we move to human viruses, but we're moving to RSV this spring. And what you see on the top is a rotating drum. So it's a really big chamber that rotates very, very, very slowly so that when we spray things in, they'll actually stay in suspension as opposed to just settling out over time. And we can change the temperature in the chamber and the humidity. Humidity is a bit hard to control, but that's another story, but we're figuring out how to do that. And then you can just sample what's in the chamber over time. You can do qPCR to say how much virus is there, and then you can do a plaque assay and try to infect cells and say how much of that is actually viable virus at the end of the day. And we can just study, hopefully, I mean, things sound like it's easy. It's been horribly not easy so far, but in a perfect world, we'd be able to come up with relationships like I'm showing here for literally for different viruses, how much will they survive as a function of temperature, whether or not there's mucins, humidity, and things like that. So to summarize, we know climate's changing, and we know that it will impact the timing of when infections happen, but there's also a lot of evidence that it could impact the magnitude of infections, which could be particularly problematic from a planning perspective. And so really what we're trying to do is come up with some first principles understanding of why that's happening, particularly for respiratory infections, although like I said, I think these would be transferable tools to think about transmission by other mechanisms as well. So my goal is to use my experimental tools to be able to come up with real relationships for, let's say, how do mucus barrier properties change with climate? How does survival in aerosols change with climate, or this breakout process of fluids that I spent a lot of time talking to you about? And then in a perfect world, we could come up with real parameters that we could plug into an SIR model or some more complex version to be able to come up with predictions that we think should be more sort of physically grounded, which would then make it easier to generalize to different places in the world and to different climate regimes as well. So I hope I'm okay on time, but I'll leave it there, and I'd be happy to take it.